Good afternoon. In last lecture, I have discussed the different classification of viruses, then the morphology of viruses. Today, we will see the next part of our syllabus, that is replication or the multiplication of human virus. Yes, today we will see the next part of our syllabus, that is the replication or the multiplication of human virus. The viral replication is the term used indicate the formation of biological viruses during the infectious process in the target host cell. Here we can see the simplified diagram of hepatitis virus replication cycle. Yes, here we can see the simplified diagram of hepatitis virus replication cycle. The viruses must first penetrate and enter the cell before viral replication can occur. Yes, the viruses must first penetrate and enter the cell before the viral replication can occur. Then, from the perspective of the virus, the purpose of viral replication is to allow the reproduction and the survival of its kind. Then, by generating the abundant copies of its genome. Yes, what? By generating the abundant copies of its genome and packaging these copies into viruses, the viruses is able to continue the infective new host. Then the replication between viruses is varied and depend upon the type of genes involved. Then the most DNA viruses assemble in the nucleus. And the most RNA viruses develop slowly in the cytoplasm. Yes, what I am saying, I am repeating here. Here, the most DNA viruses assemble in the nucleus and the most RNA viruses develop slowly in the cytoplasm. Then, viral populations do not grow through the cell division. The viral population do not grow through the cell division, then because they are acellular. Instead, they hijack the machinery and metabolism of the host cell to produce what? To produce the multiple copies of themselves, and they assemble inside the cell. Here we can see the diagram of hepatitis virus replication cycle: attachment, endocytosis, then endoplasmic reticulum, protein synthesis, and the release of attachment of the carbon. Then we will see the lytic cycle. So what is lytic cycle? The multiplication process of virulent phase is called as lytic cycle. Keep in mind, it is important for exam point of view, the replication and the multiplication, that is lytic cycle and lysogenic cycle. The multiplication process of virulent phase is called as lytic cycle because the host bacterium is lysed. Why it is called lytic cycle? Because the host bacterium is lysed at the end. There are different steps that are involved in lytic cycle. First, adsorption, then penetration, then eclipse phase, then maturation, and last, that is lysis and release of new virions. Yes, there are different steps that are involved in lytic cycle. First step, adsorption, then penetration, then third, ellipse phase, then maturation, and last, that is lysis and the release of new virions. We will see one by one. In first, adsorption, the attachment of a virus particle to the host bacterial cell. In that first, there is attachment of virus particle to the host bacterial cell. Then second, the attachment occur at the specific receptor site present at the wall of the host cell. The attachment occurs at the specific receptor site present at the wall of the host cell. Then T1 phase attached to the host with the help of tail fiber. With the help of tail fiber. Then next step, that is penetration, the injection of the nucleic acid into the host cell. Then the all of the host is hydrolyzed by the lysozyme. The all of the host is hydrolyzed by the lysozymes present at the tip of the tail. Then a hole is formed at the all of the host cell. The hole is formed at the all of the host cell, and the nucleic acid is passed through the hole into the host cell. The hole is formed at the wall of the host cell, and the nucleic acid is passed through the hole into the host cell. Then the tail sheath is contract and the central tube is pushed through the hole in the wall. Then the process is activate and uses the ATP, that is adenosine triphosphate. Then the protein coat remain attached outside the host cell wall and it is called a ghost. I am repeating the process of the step penetration. In the first, the injection of nucleic acid into the host cell. Then the wall of the host is hydrolyzed by the lysozymes present at the tip of the tail. Then a hole is formed at the wall of the host cell, and the nucleic acid is passed through the hole into the host cell. Then the tail sheath is contract and the central tube is pushed through the hole in the wall. Then the process is activate and uses the ATP, that is adenosine triphosphate. Then the protein coat remain attached outside the host cell wall and it is called as ghost. Then third phase, eclipse phase. The further infection by the same type of phase is prevented by the production of specific enzyme called as repressor. Then suppression of all host cellular activities. Then the suppression of all host cellular activities. Then the synthesis of new enzyme. Then the synthesis of new enzyme by the viral nucleic acid called early proteins. Then the viral coat protein are synthesized called as late proteins. Then maturation, the assembly of virus component into mature or the complete virion. In maturation, first the assembly of various components into the mature or the complete virions. Then head and tails are first assembled separately. Yes, here in maturation the head and tail both are assembled separately, and then they attach to form a new phage particle. Then the time between the injection of viral nucleic acid and the appearance of new phage progeny is called as eclipse period. So what is this period? That is the time between what? The time period between the injection of viral nucleic acid and the appearance of new phage progeny. This phase or this period is called as eclipse period or eclipse time. And then the total time taken from the injection of nucleic acid to the rupture of the host cell wall is called as latent period. So it's called as what? That is latent period. And then the lysis and the release of new virions. The cell wall burst at the end of latent period. The cell wall burst at the end of latent period and the virions are released. This phenomenon is called as lysis. Then the number of virions produced per host cell is specific and it is termed as a burst size. So this is all about lytic cycle. Then we will see the lysogenic cycle. Here we can see the lytic as well as lysogenic cycle of viruses. First, lytic cycle. Host bacterial cell, then bacteriophage. This is the phase infected cell. Then phase DNA, host DNA. This phase DNA circularizes the remaining separate from the host DNA. Then phase DNA replicates and phase proteins are made. New phage particles are assembled, and then the cell aligns and releasing the phage. This is all about what? That is lytic cycle. Then lysogenic cycle. The phase infects the cell, then phase DNA become incorporated into the host genome. Then the cell divides and prophage DNA is passed onto daughter cells. Then under stressful conditions, the phage DNA is excised from the bacterial chromosome and enters the lytic cycle. The lysogenic cycle is associated the lysogenic cycle is associated with replication of temperate phage, that is prophage. In this type of cycle, the viral genome become associated with host genome. Yes, in this type of cycle, the viral genome is become associated with host genome. This phenomenon is called as lysogeny, and the cycle of the temperate phase is called as lysogenic cycle. The bacterium which harbors the phage is called as lysogenic bacterium. Then the phage DNA is replicated along with the bacterial genome. Then the UV radiation and the presence of hydrogen peroxide. Yes, presence of UV radiation and the hydrogen peroxide, it may causes the excision of the prophage from the bacterial genome and convert into virulent stage, and this process of conversion is called as induction. Next point, that is cultivation of viruses. The viruses are, as you know, the viruses are obligate intracellular parasite, so they depend upon host for their survival. They cannot be grown in non-living culture media or on agar plate alone. They must require living cell to support their replication, as I said earlier. The primary purpose of viral cultivation is first. So what is the purpose of cultivation, or what is the purpose of virus cultivation? First purpose, to isolate and identify the viruses in clinical sample. Then second, to do the research on viral structure, then viral replication, genetics, and the effect on the host cell. Then to prepare viruses for vaccine. Then the cultivation of viruses is essential, which is carried out by three methods. First, animal inoculation, then inoculation into embryonated egg, and the cell culture. There are three methods that are categorized under the cultivation of viruses. First method, animal inoculation, then inoculation into embryonated eggs, and the cell culture. In cell culture, it is again divided into cell line culture, explant culture. Here we can see the tree diagram. Cell culture, then in cell culture it is again divided into cell line culture, then explant culture, and organ culture. In cell line culture, the primary cell line, semi-continuous cell line, and the continuous cell. We will see the animal inoculation. The viruses which are not cultivated in embryonated eggs and the tissue culture, these are cultivated in laboratory animals. See here what I am saying. The viruses which are not cultivated in embryonated eggs and the tissue culture, that are cultivated in laboratory animals such as mice, guinea pig, hamster, rabbit, and primates are used. The selected animals should be healthy and free from any communicable disease. Yes, these selected animals, it should be free from or it should be healthy and should be free from any communicable diseases. Then suckling mice of age less than 48 hours are most commonly used. Then suckling mice are susceptible to toga virus and Coxsackie viruses, which are inoculated by intracerebral and the intranasal route. Yes, which are inoculated by cerebral and the intranasal route. Then viruses can also be inoculated by intraperitoneal and the subcutaneous route. Intraperitoneal and the subcutaneous route. Then after inoculation, viruses multiply in host and develops a disease. The animals are observed for symptoms of disease and the death. Then the virus is isolated and purified from tissue of this animal. Then the live inoculation was first used on the human volunteer. Yes, what I am saying here, the live inoculation was first used on human volunteers for the study of yellow fever viruses or the yellow fever virus. Then, so what are the advantages of animal inoculation and what are the disadvantages of this animal inoculation? First, we will see the advantages of this animal inoculation. First advantage, diagnosis, pathogenesis, and the clinical symptoms are determined. Second advantage, the production of antibodies can be identified. Then, primary isolation of certain viruses. Then, mice provide a reliable model for studying viral replication. Then, next, the used for the study of immune response and epidemiology and also oncogenesis. Next, that is disadvantages of animal inoculation. First disadvantage, expensive. Then second, difficulty in maintenance of animal. Then difficulty in choosing the animals for particular viruses. Then some human viruses cannot be grown in animal, or it can be grown but do not cause a disease. Then next disadvantage, mice do not provide model for vaccine development. Then next disadvantage, it will lead to generation of escape mutants. Next, the issues related to animal welfare system. So this is all about the lytic cycle, lysogenic cycle, and the some part of cultivation of viruses. Next part we will see in next lecture.